All right, our next unit in AP biology, as you can see, is heredity. Um, we've spent a lot of time now talking about how cells work, and the basic idea is that life is all about breaking down molecules to get raw materials and enough energy to build new molecules that will form your body and proteins that can carry out functions. The primary function of that would be to respond to the environment via cell communication pathways. And what you get is this huge, chaotic molecular dance of interactions that leads to an emergent property called life. But one of the things that seems true about life is that eventually the entropy of the universe is going to win, that you're fighting against that chaos using energy, but eventually the energy is going to run out and life is going to stop. But while you can't live forever, you can give your life to another organism via reproduction. So on the screen here, you have what looks like 4 generations of chickens as they pass their traits throughout the generations. And the unit on heredity is all about how does this happen? Um, and when you say you give your life to your offspring, what are you actually giving? What you're giving is information. And specifically, you are giving chromosomes, right? So here's where the story kind of begins, is that we have to give chromosomes to our offspring. Now you have, and most organisms have 2 sets of DNA. You are a diploid organism, which is an important word. There are some organisms that aren't diploid. Of course, prokaryotes, to remind you, being the most prominent group in that they only have one singular circular chromosome. There are some plants that have 3 copies. They are triploid, but most things you're going to encounter are diploid. This means that you have 2 of every single chromosome, and on those chromosomes are genes, but there are different versions of genes. So sometimes you can get different versions, and here's where we get into these things like homozygous, where you have 2 of the same uh, version of the gene, or you can be heterozygous, where you have 1 of each version of the gene, but you always have 2. This is called your genotype, and this is what is going to give the information to your proteins for how your body is going to be run. But when we're talking about reproduction, the biggest key here is that you can only give 1 of each chromosome, right? You have 2 copies, but you can only give 1. And why can you only give 1? Because if you give 2 copies, then the numbers get messed up, right? You need to combine your information with that from another organism. And so if you both give your diploid cells, then the offspring would have 4 copies of their chromosomes, which would be a bad thing and not lead to a living organism. So we need to make special cells called gametes that are haploid down there. Notice the N plus N. Makes 2N. And so that way we cut our DNA information in half, then we combine that half with another half to make a whole again. And this is the good old cycle of life. And so what we want to know is how that happens. And it's by something called meiosis, which happens in 2 steps. Now, this is very closely related to mitosis, which there's a whole video just on that. So I'm not going to go through all that here. It'll just be about the differences. And the #1 difference between meiosis and mitosis is that we want to separate homologous chromosomes. The goal is to get these copies away from each other, right? You got, out of these 2 #2s that the arrows are pointing at, 1 of them came from your father, 1 came from your mother, and you can only give 1 of them to your kid, right? So we got to get those apart. We got to separate these things. And so the first step that makes this different from mitosis is that the chromosomes are going to pair up, right? The 2 #1s, the 1 from dad and 1 from mom, are going to get next to each other. And then they are going to line up as pairs. And so here's one of the most important shots on the whole part, is that you see that imaginary dotted line, and that there are 2 big ones here at the top. That 1 is on each side of the imaginary line. And so when they split during anaphase, this one goes that way and that one goes that way. And you end up with the 2 big ones separate from each other. And you can see there's also 2 smaller ones that are away from each other. And that's called separated homologous chromosomes. It goes through the good old prophase, metaphase, anaphase, telophase, but they line up together as pairs. Then, once they are separate into from their homologous pairs, they're now haploid. This is the N part. Now you notice that they line up the same way they do in mitosis during the second part, because mitosis is all about separating sister chromatids, which is exactly what we're doing here. So a lot of times they'll just say meiosis is where you do that special version of mitosis, and then you just do the regular version of mitosis right after it. The big key here is that the cells that you end up with are haploid. They have half as many chromosomes as what the original parent cell started with. These are not duplicates. These are not clones. And in fact, the whole point of this is to make variation. You don't really want them to be clones because we want to start shuffling the chromosomes up as eventually we learn about in evolution that variety is good. And so one of the biggest parts about the meiosis section was not just the mechanism, but where does the variation come from? And the first way it happens is something called crossing over, where the homologous pairs will trade corresponding segments during prophase 1. Now, the important thing here is that when you look at, say, this gene or this chromosome right here, let's say that the A and B are for hair color and eye color, and that the big A, big B give you brown hair and brown eyes. If this chromosome was given to the kid, then you would expect it to have brown hair and brown eyes. But down here, you have the little a, little b. Let's say that's for blue eyes and blonde hair. Then you would get a kid, if they got that chromosome, that they would have those traits. But thanks to crossing over, you now have an option to have brown hair and blue eyes and brown eyes and blonde hair, right? Like those are two combinations that didn't exist before. And so by crossing over, we get all these new combinations of genes on chromosomes, which we can then shuffle up and give to our kids. The really where a lot of the variation comes from is the shuffling, and that's called independent assortment, meaning that the chromosome 1 doesn't care what chromosome 2 is doing, and so they can line up independently of each other. This is specifically during metaphase 1. Here you have the pictures that show you that in this version, the blue and the yellow end up on the same side, and that makes them get into cells where there's blue and yellow, and their final products are blue and yellow. Whereas in the bottom picture, the blue and the blue go on the same side, and the yellow and yellow go on the same side, so you end up with all blue and all yellow, right? And so by lining up independently, this one right here doesn't know what this one is doing. Sometimes they end up together, sometimes they don't. And this is going to shuffle things up and create a lot of randomness. You might remember that makes 8 million possible sperms and eggs in humans. The final step of creating variation isn't really about meiosis, but it's just the practicality of now that you cut everything in half, you have to combine them again. And now you have 8 million possible combinations with another 8 million possible combinations. And something that we'll keep on pointing out during this section is that when you have 8 million sperm and eggs, that and means multiply, and so that means a whole lot of combinations. Plus then you have the crossing over that makes it even more, right? And so now we're ready to get back to this part, right? Now we know why you can only give one and how each one is given. But genetics is a lot about predicting things. And so if you have 2 chromosomes and you have to give one of each, then the chances of you giving those things are 50%, right? And so if I have a 50% chance of giving my chromosome and my spouse has the 50% chance of giving their chromosomes, basically every time you make a kid, you have a bunch of 50-50 chances being multiplied times each other. And on the simplest version, this gives you 4 possible combinations of giving say this big A with this little a, or the big A with the big A, right? And so here's our Punnett squares. Now I'm not going to go into all the Punnett squares. This is usually the strongest part of everybody's genetics game. But you can see that every possible combination of mother and father chromosomes, in case you were wondering, that says father right there, right? The mother in one case has a 100% chance of giving a big A, but sometimes has a 50-50 chance of giving things, right? This parent has a couple more 50-50 chances. You could do this mathematically. You can draw it out using the Punnett squares. And there will actually be another video where I'll go a little bit more into the math because I don't want to make this one too super long, right? But the idea is that this is your genotype. And if you know your genotype, you can figure out your phenotype, which is what you end up looking like, what your actual trait is. This is where the idea of dominant and recessive comes in, that genotypes can be different, like these two round peas. But because the round gene is dominant, it covers up that recessive wrinkled gene. And so you see round twice, but it's not actually happening twice on the genetic level. You have to interpret the genotype into the phenotype. Um, Recessive ones are super important. We ended up not having to do the evolution part on the AP exam, but if you remember, recessives give us a lot of information because if you see someone with a recessive phenotype, there's only one possible genotype, which is to have the two recessive genes. And so when you're trying to do like figure out puzzles and things, that's very helpful. So the trick about genetics is that there's a lot of different levels at which the genotype determines your phenotype. The most simple is the single gene traits, and that's usually where questions are going to come from. These are things that are relatively simple in their mechanism. There's a number of genetic diseases like cystic fibrosis and sickle cell anemia that we talked about, where one gene can really impact your entire phenotype. But those are rare compared to polygenic traits. Most traits for humans, especially that are very complicated organisms, have many genes that control them. And so this makes it a lot harder to predict. Finally, the environment also comes into play, and this makes things even more difficult as we try to predict if you have these genes, what will you end up like? Well, it also depends on like where you grew up and things like that. And so I want to briefly talk about these polygenic traits, or a little bit more in detail, I guess I would say. And it's all about this bell curve right here, that in a polygenic trait, there's more than one gene. In this particular example, it's using skin color as a basic model for where you have three different genes, the A, the B, and the C gene, and there's a dominant and recessive version. And so in the end, you're just doing a lot of Punnett squares, but the dominant versions stack on each other. Right? So if you have one copy of the dominant gene, then you have a little bit of coloration in your skin. If you have two copies, then you have even more. If you have three copies and four copies, right? And you can see as you go along. And so when you get two heterozygous individuals, which is what this picture is showing you, and you can see on this one a little bit better where it basically shows this gigantic Punnett square, there's 1 out of 64 chance of being this guy right there, and another 1 in 64 chance of being this one right there. But when you get towards the middle, there's more and more chances. And so in the end, you get this spectrum. This is an important idea, that there aren't usually these discrete groups of, say, sickle cell versus not sickle cell, right? It would be, well, you're kind of short and you're a little bit taller and you're a little bit taller and you have this huge range of heights or skin colors or whatever else that might be. So those don't follow the normal Punnett squares because they get a lot more complicated. Again, the environment I was mentioning, we know that a lot of these traits are linked to genes, but your lifestyle is important. If you have a history of cancer in your family and then you go and smoke a bunch of cigarettes, that's probably going to be a bad thing. Obviously, you could get cancer even if you didn't have the risk, but the risks go up based on your genes. But let's say you do have that risk from your family of having the genes for cancer, then if you don't smoke, then you're going to help lower your chances of getting cancer, right? So it's a combination of your genes and your choices. There are some examples of where the environment just straight up affects things. Malnutrition during childhood leads to you being shorter than you would have been normally. Your environment where you grew up basically changed how tall you were going to be. The Himalayan rabbits down here, their fur color is dependent upon the temperature at the time when the hair is growing. So they shave the rabbit and they put a cold on it and the fur color, or the fur comes back darker, right? I forget which kind of flower this is, but the color of the flowers depends on the pH of the soil, and you can manipulate this and create flowers whatever color you want within that spectrum, right? And so there's a lot of different versions. You don't have to memorize any of these, but there's a lot of different ways that the environment can affect your phenotype, and these things again are going to change the genetics. It's not going to be as simple like, oh, are you a big A or a little A? Like you've got to now you have to factor in these other things. Um, Getting more back to Punnett squares, X-linked traits are something that violate Mendel's laws because in this case, your percentages change whether you're a male or a female. Males only have one X chromosome plus the Y chromosome. So if there's any gene on the X chromosome that has a mutation, the male only needs one mutation to see the effect, whereas the female still needs the normal two mutations to see the effect. You can see in the diagram on the right, um, it affects your gender because the male always gives his Y chromosome to his sons. So in this case, when the male is unaffected on his Y chromosome, he gives that to his sons. That's not from him, right? Like that's going to end up um, the boy that ends up affected, he doesn't get it from dad, he gets it from mom, because mom is the one that has, and I don't know why that's happening, of course. Mom's the one that has the mutated X. Mom has to give her X to the boy because the boy's got to get at least one X chromosome. And now the boy has the good Y from dad, but it's meaningless, and the bad X from mom, and now he has that particular trait. And so this affects your Punnett squares, right? And so it's something to keep in mind. This is a very common example that they use on the test. Note the way that the genotypes are written so that when you do your Punnett squares, it's helpful to keep track of the X's, Y's, and big or little A's or whatever letter it happens to be. Aside from sex-linked traits, there's a couple of versions of, I don't know where to put my face, of inheritance that don't follow the normal dominant recessive. You can see them on the screen. Incomplete dominance, like when you have these carnations where a big R and a little r mix together to make pink, right? Incomplete dominance, the red eye, think of it as incompletely covering up the white. It covers up the whites mostly, but it's still there a little bit, and so it ends up pink, and so they blend together. The other version called codominance is when they don't blend together, where they are seen independently. So the red horse and the white horse make this roan horse that has red hairs and white hairs because some of the hairs are expressing the big R allele, and then some of the hairs are using the little r, and so it changes the color. There's no pink on that horse. And here's a flower that's showing codominance, where before, instead of the red and white making a pink, it made a red and a white flower. The last one, again, I only show this one because it's very common as an example, and it's a great example of dominant recessive and codominance, because in the blood types, there's 3 different versions. There's the A, B, and O, and A and B are kind of independent of each other. A makes the green spikes, B makes the blue circles, but O is the mutant version. O is the broken version that makes nothing. And so right off the bat, A and B are dominant to the O, because if you get an A and an O, well, then you have green spikes plus nothing still makes green spikes. Or if you have the B and the O, blue spikes plus nothing makes blue spikes. But because the A and the B are independent, if you have an A and a B, you have green spikes and blue circles, because they don't blend together. They don't cover each other up. They're expressed both at the same time, right? And so again, that's the great example of how the idea of dominant and recessive or codominant is really based on the mechanism of the molecules and what the molecules are doing as they go to create the phenotype. So this one, the codominance and the incomplete dominance, I find, I don't want to say aren't going to be on the test, but those ones don't get brought up a whole lot. The sex-linked is something to think about. But then there's other things that have nothing to do with Punnett squares. The main one being non-nuclear inheritance, meaning that your mitochondria and chloroplasts, not for you, of course, but for plants, the chloroplasts, they have their own DNA, and mom always gives the mitochondria to the child. The egg has the mitochondria in it. The sperm has basically just a bunch of DNA. And so fathers don't pass their mitochondrial DNA to their children. And so in this case, it wouldn't matter what your Punnett square is. If your mother has a disease, say, that is related to a faulty mitochondria, her children would get that automatically because you get your mitochondria from mom, right? And so again, there's no Punnett square for doing that. It just happens that way. There are these huge chromosomal mutations that we don't really talk about when we do the mutations. you could see the examples on here of just deleting whole sections, swapping sections, reversing sections, moving sections. Sometimes they swap identically, like where they trade identical parts, but sometimes they swap not identically. And so you get all kinds of crazy things when you put a bunch of chemicals together and let them do what they do and react, right? And so these will be things that you have there's no predicting them. Unfortunately, these are just things that happen, and then there's not really any Punnett square necessary. Finally, that leads to one last example of something that kind of follows Mendel, but it doesn't, because it has to do with mitosis. It has to do with trisomy number 21, which is having 3 copies of, in this case, chromosome number 21, which leads to Down syndrome. Down syndrome is not something that you really inherit. You do inherit it, but you don't inherit it like from a Punnett square. You can't predict it. It's from a random mistake that happens during meiosis. It's called non-disjunction, meaning that the pairs failed to separate. It can happen in 2 different places because there's 2 different times where the chromosomes separate. Let's say that you have your chromosomes here during meiosis 1, and they're in metaphase because they're lined up in pairs, and then they split. But they didn't split. The number 1 chromosome split, the 21s didn't. The cell doesn't really realize that this is happening. It can't really make the distinction. So it just keeps going, and it goes to metaphase 2, where they line up in a single file line, and then you split those during anaphase 2, of course. And that all happens normally. But 2 of the sperms or eggs come out with an extra chromosome, and 1 of them comes out, or 2 of them come without 1. You can see down there, right? And so if this egg or sperm gets used to make the kid, then that kid's going to end up with an extra chromosome number 21, and that kid would have Down syndrome. If you were to make the baby with the one that's missing the 21, it would probably just end in a miscarriage because the developing embryo would have too many problems with their genes. The second way this could happen was if it separates normally during metaphase 1, but then when metaphase 2, this one didn't separate. And so in this case, you get 2 eggs or sperm that are normal, but there's 1 of each version of the messed up one. And of course, again, if you use the one, if this happens to be the one random one that makes it to the egg, then you have your Down syndrome. And again, these are things that are called non-Mendelian because you can't predict them using a Punnett square. It's just random things that happen during development. And so those are the basics of heredity. Again, there's a little bit more math and some things that need to happen, but I'm pretty sure I've been talking for a while already. And on this one in particular, if you're looking for extra practice problems, we could probably dig those out. And so just let me know. Otherwise, good luck on the progress checks.